So thank you, Tola, and thank you for inviting me to show you the things that we do at the lab at Tel Aviv University. So we're working on tissue engineering, and we're engineering different types of tissues from the heart, brain, spinal cord, intestine, kidneys, and other tissues. And I'll give an overview of what we do in the lab. So when we talk about tissue engineering, about the concept of tissue engineering, so oops, it takes time. So we have a patient with a defective organ, as you can see here in this slide. And what we do in the lab, we're engineering a substitute tissue that would have the same function as the natural tissue. We then remove the defective tissue, and as you can see, under sterile conditions, we take the engineered tissue and we transplant it so that the organ regains function. And when we talk about tissue engineering, when we really look into it, so what we do, we take cells, preferably from the patient, because we do not want to provoke an immune response. And initially, we're expanding the cells in the lab in 2D on flasks or Petri dishes. And then we need to create tissues. And tissues in the body are not just the cells. The cells are extremely important, but the biomaterial in between the cells, the extracellular matrix, is important as well. It directs the cells, it gives signals to the cells. And what we do in the lab, we're developing different types of biomaterials so that we can take the cells, seed them within. We can add growth factors or particles, slowly releasing factors, small molecules, nanoparticles. And essentially, we're doing everything we can to allow the cells to assemble into a functioning tissue, which can then be taken and transplanted on the defective organ. And when we talk about cardiac tissue, and this is the case for different tissues as well, tissues that we cannot actually take cells from the body. With the heart cells, cardiac cells, we cannot take cells from the heart. We cannot expand them because these cells do not divide. So we need to use other cells, stem cells, but the stem cells that we do have in our body are not capable of differentiating to heart cells or neurons. So what we can do, either use embryonic stem cells, which would provoke an immune response after transplantation, or we can induce pluripotent stem cells by reprogramming the cells. So this is what we do in the lab. We reprogram the cells to become iPSCs. And then we can differentiate the cells to any cell type that we wish. So if we're talking about the heart, we can create fibroblasts as a connective tissue, cardiomyocytes and subtypes of the cardiomyocytes, smooth muscle cells and endothelial cells for the blood vessels. And we can create different types of tissues. So we can create 2D constructs. These are mainly good for drug screening. Or we can create 3D vascularized cardiac patches to regenerate the left ventricle of the heart. Or we can start even playing with engineering the entire heart. And I'll explain what we do. So when we talk about biomaterials for tissue engineering, there are a few forms of materials that people use. So we can use hydrogels. You can see here, hyaluronic acid hydrogel. What we do with that, we can mix the cells within, and this can be injected or transplanted. We can create preformed scaffolds, some kind of sponges with pores. We can seed the cells into these pores and then transplant these scaffolds in the cells. Or we can create fibrous scaffolds, which the biomaterial really mimic the morphology of the extracellular matrix, as you can see, in this case, PLGA fibers by electrospinning. So we can use either type of, either form of materials. And initially, when we started working on heart patches, we wanted to evaluate first how the extracellular matrix of the heart really looks like. And we took pig's hearts. We removed all the cells, we cleaned them, we cleaned the tissue. And then we went to see the fibers in the SEM. And what we could see is one big mass of fibers. We could see microfibers and nanofibers, randomly oriented fibers, aligned fibers. And initially, we wanted to evaluate what's the role of the different fiber populations. So synthetically, we created the microfibers or nanofibers, randomly oriented ones or the aligned ones. And I'll give you two examples of how the cells behave differently. You can see on the randomly oriented fibers, you can see that there is no anisotropy of the in the cells, and with the aligned fibers, the cells are aligned. And this is extremely important when we talk about cardiac tissues, because we want the cells to propagate the electrical signal nicely from one spot to another. Without the alignment, this cannot be done. So this is one example how the morphology can change the outcome of the tissue. Another one is the effect of fiber diameter. So we created the nanofibers, 1 micrometer fibers or 5 micrometer fibers in diameter. And at least in Israel, 10 years ago, when you mentioned the word nano, it was a buzzword. If you put it in any grant, you increase the chances of acceptance at least in 50%. However, cardiomyocytes don't really like the nanofibers. They prefer the bigger fibers. You can see here, they're rounded on the nanofibers, and when they have more place to adhere to, like the bigger fibers, they're more elongated, and they have more pronounced sarcomeres, the contracting units of the heart. And this is another example of how you fabricate biomaterials, and you basically change the outcome of the tissue. So if you take into consideration the morphology of the fibers, the mechanical properties, which I didn't talk about, or the biochemical content of the biomaterials, you can create such cardiac patches. This is few centimeters in diameter, few millimeters in thickness, and these cardiac patches, they contract nicely in the lab without external stimulation for a long period of time. You can take these patches and transplant them on the infarcted heart of rats, and we saw really nice regeneration. However, when we talk about hearts, human hearts, we're talking about something that is much thicker, about 1 centimeter thick tissues. And when we go into thick tissues and we try to seed the cells, we have mass transfer limitations. Oxygen cannot penetrate deep into the tissue. And most of the cells in the core of the tissue simply die. And what we've decided to do was to create a different approach. We call it the bottom-up approach to assemble thick cardiac patches. And what we do here, we take electrospun fibers. We use lasers to pattern, for example, grooves for cardiomyocytes to have aligned tissues. We have many of these thin tissues. We can have another layer or a few layers of channels for endothelial cells and honeycomb structures for VEGF releasing particles to promote vascularization. And we have honeycomb structures for dexamethasone releasing particles to avoid rejection. So we basically culture these thin layers separately in the lab. And then just prior to transplantation, we assemble thick tissue about 1 centimeter thick. And this is one example showing that once we release VEGF from these particles within, we can have nice penetration of blood vessels. And this thick tissue is beautifully vascularized and viable. So we have these biomaterials that are suitable for, this approach to create thick patches. But when we talk about patches that will not provoke an immune response, and if we use the synthetic materials surely promote an immune response once we transplant them. Sometimes it's not critical, but when we talk about tissues that need to transfer electrical signal, if we have a fibrotic tissue around them, the signal stops, and this is not relevant. We're talking about cardiac tissues or neuronal tissues. So people thought, let's use materials that come from animals. This should not provoke an immune response. And people took, for example, pigs materials, decellularized them. And there are millions of people who already received these materials in the clinic. However, they also receive immunosuppressors to avoid rejection. And what we were able to show first that there are certain antigens, for example, that humans do not have and pigs have, like this alpha-gal sugar, and this provokes an immune response. And also the ECM proteins that we do share with pigs, for example, their amino acid sequence is completely different, and the protein folds differently and provokes an immune response. So what we've decided to do was to rely on the patient's own materials and the patient's own cells. And how can we do that? We can take fatty tissue from the patient, we isolate the cells, we reprogram the cells to become induced pluripotent stem cells, and the biomaterial of this fatty tissue is processed to become a porous scaffold. We can take the cells, see them within, differentiate to any cell type, and we have completely autologous tissue that will not provoke an immune response. And if we look how the cells are seated and behave within, so you can see that they are aligned, and we can create nice vascularization within these patient-specific scaffolds, and they contract nicely. And if we look at the immune response to these materials, what we've done here, we simulated the immune response in a dish, human immune response in a dish. So we took donors' blood and donors' fatty tissue, and we isolated the antigen-presenting cells from the T cells. We incubated these antigen-presenting cells with the materials either from pigs or from the same patient or from other patients. And after 2 days, we've added the T cells, and we could see two nice things. First of all, that there is a difference, a significant difference between the pigs materials and the human materials, but also that there is a difference between the donors. So I react differently to other people's materials than other people to mine. So there is a need for patient-specific material as well, not just the patient-specific cells. And we've shown it also in animals. So now we have patches completely autologous. We can take them, transplant them on the infarcted heart. But what you can see from this movie, that two things. First of all, there's an open chest surgery here for these cardiac patches, which is not ideal for patients who just had a heart attack. And another thing that you can see is that there are stitches to secure the patches to the heart, which is not ideal as well. And we'll talk about the stitches in a few slides. To avoid the need for an open chest surgery, what we've done, we created thermoresponsive hydrogel from the patient's own materials. So we mill the material, we process it to become liquid in room temperature. Now we can mix the cells here, and in 37 degrees, it solidifies. So now we can bring this material into the heart, into the brain, or wherever we want, and we can form the tissue inside the body. So this is an overview of what we can do. So we take this fatty tissue, we isolate the stromal cells, we reprogram them to become induced pluripotent stem cells, and the ECM is processed to a personalized hydrogel, and we can create any tissue that we wish. We just mimic the embryonic development of different tissues. For example, we can create adipogenic tissues for reconstructive surgeries, motor neuron tissues for spinal cord injuries, cortical implants for brain trauma or stroke, dopaminergic implants for Parkinson's disease, and cardiovascular implants as well for cardiovascular diseases. And we can use different, more advanced technologies such as 3D printing and microfluidics to create more complex tissues. And based on that, we formed a company, Matrisoft, which hopefully in the next 6 months will be in clinical trials with spinal cord implants. So going back to the heart, so these cells used to be fat cells, and then induced pluripotent stem cells, and after differentiation within the hydrogel, in 3D, they became cardiomyocytes. You can see that they have markers of heart tissues, and they contract. They transfer the electrical signal And once you take them and transplant them on the infarcted heart, you can see that we can elevate the ejection fraction in small animals, as well as in pigs. We can inject them into the heart. Of course, this is an open chest surgery in pigs, just because it is expensive to do it without opening the chest, but we can do it from the inside as well, not just, and we can elevate the ejection fraction in pigs as well. So now we have this hydrogel with the cells. So we can use it also to create more complex tissues, and we can use 3D printing to create these tissues. So what we do here, we look at the left ventricle of a patient. We look at the area where the infarct is. You can see it here. We look at the blood vessels. We transfer these blood vessels to the computer. And then we need to assure that these blood vessels, since we cannot see all the small scale blood vessels in the CT, so we need to do mathematical modeling to ensure that these patches are getting oxygens throughout them. And once we do that, we go to one of the printers in the lab, and we're printing the patient's own cells with the patient's own materials. So we print the parenchymal tissue, the cardiac tissue, basically, and the blood vessels, endothelial cells, smooth muscle cells, fibroblasts to support the cells. And eventually we're getting a patient-specific printed patch, which fits the basically the anatomy of the patient, the biochemical profile of the patient, and also the cellular profile of the patient. You can see here a cross-section of one of the blood vessels. The resolution that we can get for these blood vessels is about 100 micrometer in diameter. I'll explain later what we do if we want to create to control capillaries. But we can see, and you can see it in pink, that after we create or print the large-scale capillaries, they're sprouting out of the small-scale capillaries to the entire patch. If we look at the function of the patch, you can see that there is a nice contraction. This is the blood vessel, the entrance to the vessel. And basically, we can also not just use it for regenerative medicine, but we can also use it to test drugs, and test drugs not in normal organ-on-a-chip technologies, because now we can do it in 3D, perfuse the drugs throughout the blood vessels, see how these drugs go out of penetrate throughout the blood vessels and affect the tissue. And this is a collaboration with Bayer. We're showing that we can do it using this technology. So now I've mentioned that we do not want to have an open chest surgery. However, if we print the tissues, we need to open the patient because the patches are big. And to avoid that, what we've done, we printed the tissue in the soft material. And after the tissue is already functional, we can see the contraction in the lab, we can add a small molecule, oxidized sucrose, as you can see in B. Basically what it does, it cross-links a little bit the tissue, and it provides really good mechanical properties that we can now fold the tissue, put it into a trocar, and then bring it into the heart with a minimal invasive technology. And I'll skip that movie, but you can see before and after injection, this was, we did it up to 6 centimeter cardiac patch. We were able to show that before and after injection, we have these patches are still functional. So we're printing tissues, but these are, we call it in the lab 2.5D tissues because they're not really volumetric tissues. If we want to create real volumetric tissues, we need another approach because we cannot print this liquid high or a few layers because it will collapse. And what we've done here, we developed a way to have a lot of nanoparticles within this glass cup. And the needle from the printer goes through these particles, deposit the material, the particles, all the liquid in the liquid or the collagen fibers, the patient's own material in place. And after it is heated to 37 degrees and solidifies, we can add another molecule that will remove or dissociate the nanoparticles, and we're left only with the printed tissue. And with that, we were able to print nice structures like that. I really like that one over there. It's a ball within a ball, and it solidifies in 37 degrees, and you can see, and it is made of the patient's own materials and cells and everything. So this allowed us in 2019 to print the first small-scale human heart with the blood vessels. So everything came from the patient. We had large blood vessels, small blood vessels within these hearts, and you can see here in the middle of this process and the end process, we can have these hearts, and the cells within contracted, but they did not contract synchronously. And what we needed to do next was to mature or teach them how to speak with each other on the electrical level. So we did it with bioreactors. We hang these hearts, we perfused them, and provided electrical stimulation. We also took these hearts and ectotopically transplanted them into animals in parallel to the heart. We didn't want to replace the heart. We just wanted to mature these hearts inside the animal, to use the animal as a bioreactor. And what you can see here, what happens after 7 days in the animal, the entire heart contracts nicely when it is explanted, and you can look at, can see the electrical signal going through this heart. So now we have these hearts, but they're made of a mixed population of heart cells because we did not control the differentiation process to cardiomyocytes. So what we decided to do next was to differentiate to different cell types, to the atrial cells, to the ventricular cells, the pacemaker cells. And initially we wanted to first evaluate whether the pacemaker cells can control the contraction. And that we've done with printed rings. So here you can see without the pacemaker cells. Each time the signal starts from a different spot, you can see the arrows. When we use the pacemaker cells, each time it starts from the same place where we printed the pacemaker cells. And this allowed us to move forward to print the entire heart with the pacemaker cells in the right place, the atrial cells, the ventricular cells, the blood vessel cells within. So now we have hearts that contract nicely in the lab. We did some echocardiography, and we were able to see nice ejection fraction of these hearts. Still not enough to replace the heart, but it gets to between 16 to 20% ejection fraction, which is relatively high. So we have these hearts with the blood vessels, and we wanted to basically, before that, I'll say that we also print large blood vessels, and here we developed another way to print them. So we print half vessels and then the other half, and with the connectors, and it allows us to use these blood vessels as Lego, just to basically connect them with other blood vessels to create whatever structure that we want. And we're now, we transplanted it in animals. But when we talk about the small scale capillaries, as I mentioned, we currently cannot control the printing of these capillaries because there are limitations for the printers. The printers can only print about 100 micrometer blood vessels in diameter. And what we've decided to do is to use 4D printing to print the blood vessels in as 100 micrometer in diameter, and then to trigger them to shrink and collapse and to form these small scale capillaries. And for that, what we've done, we developed another bioink. So we have the regular bioink for the cardiomyocytes. We have regular bioink for the blood vessels, for the normal blood vessels, but we also have the bioink for the shrinking blood vessels. You can see it is made of the patient's own materials, the collagen fibers of the patient, and we also put nanoparticles, polynipam nanoparticles into these fibers, and we can control the direction of shrinkage, as you can see here by the printing direction. And eventually, if you look at this movie, and you can see the temperature here in 37 degrees, these blood vessels simply shrink, and they shrink 13-fold of their initial diameter, allowing us to reach 5 micrometer in diameter. So you can see in 37 degrees, it simply shrinks. So we talked about the heart. We'll get back to the heart soon, but we can also use the same technology to create spinal cord implants. So we take this fatty tissue, we reprogram the cells, we process the ECM, and now we mimic the embryonic development of the spinal cord or the motor neurons. And after a while, we're transplanting them. So here you can see after months of differentiation in 3D, we get nice 3D networks of spinal cord motor neurons. And after transplantation, so what we've done first, we tried it in 2 models. First, we did an acute model of spinal cord injury. We cut the spinal cord and immediately implanted these tissues. It worked nicely. 100% of the animals worked beautifully after 2 weeks already. But it's relatively easy to use it in an acute model because there's spontaneous regeneration as well. So what we decided to do was to create the incision, create the injury, wait enough time, which is equivalent to more than a year in humans, and only then transplant these engineered tissues. And you can see by this MRI, this is our group, the implant. You can see that there is nice regeneration of the tracts here. And if we look at the animals and how they walk, this is the untreated animal. You can see that one of the legs is not operating. And with the full implant, you can see that they walk perfectly, and 80% of the animals walk perfectly. And based on that, in 2019, we formed Matriself, and as I mentioned, we're now moving to clinical trials with the same procedure. This is just quantification. We did some usability as well in large animals. This was in collaboration with Johns Hopkins. You can see the implants in the spinal cord of the pigs, and you can see by this MRI that they feel the incision site, and we have nice regeneration. Sometimes we do not need to have big tissues. Sometimes we just need small particles, but we don't want to just inject the cells. The cells without a supporting biomaterial will not work properly. So what we do, we can use microfluidics to create this encapsulation. So we inject the hydrogel in its liquid form through the microfluidics, through oil. We create emulsions, and eventually we get many tiny droplets like that. Then we can differentiate them. For example, we did some mathematical modeling to understand what's the best dimensions for these particles or droplets. And here you can see dopaminergic implants, particles that with dopaminergic neurons that secrete dopamine. This is for Parkinson's disease. After this is 90 days in culture, they're ready for transplantation, and this is what we're doing right now in the lab. We also do cortical implants for brain trauma and stroke. The same approach. We just mimic the embryonic development of the cortex. And here you can see some of these implants and how they interact with each other. See, they transfer the electrical signal and they activate each other. Okay, back to the heart. This is work that started about 15 years ago about incorporating materials into scaffolding or nanoparticles into scaffolding materials to enhance the electrical signal of or to enhance the conductivity of the biomaterials. So we have cells in different pores of the materials, and you can see these cells and these cells, they contract, but they're not speaking with each other on the electrical level. And what we've decided to do is to incorporate gold nanowires into the walls of the biomaterials to allow the cells to interact with each other. And in a series of papers we've shown, you can see these two movies that with the gold nanowires in this case, we had a very nice conduction or transfer of the electrical signal. And in this graph, you can see that we also took the patient-specific material, incorporated gold nanoparticles, in this case, not nanowires, and by this IV curve, you can see that they turn out to be fully conducting materials, and these are natural materials. It also works nicely with neurons, so we have the fibers of the patient-specific materials, and you can see here after 3 days in culture, how neurons look like. This is a single neuron, either without gold nanoparticles or with, and you can see the extensions of this neuron. This is extremely important when we talk about regenerating the spinal cord or the brain. We need to have long neurons. In addition to better electrical coupling of cells, we want to exploit these gold nanoparticles. In this case, it was gold nanoparticles. We want to absorb reactive oxygen species after infarction, and also to use these particles to image the treatment. So when we inject it, we want to see where we injected it in comparison to the scar tissue. And this is what we've done. We have better contraction with these gold nanoparticles. We can reduce reactive oxygen species because they adhere or absorb to the gold particles. We can image the treatment, and basically we can improve heart function after infarction. Already after 2 weeks, we can see that it works. If you remember, I talked about the stitches to the heart, which are not ideal. And in this case, what we decided to do was to rely on the physical properties of gold nanorods, in this case. And we incorporated gold nanorods into the biomaterials. And the nice thing about these nanorods that if you illuminate them in near IR, they convert the energy to thermal energy, which we can solder the tissue into the heart without the need for stitches. And this is after, you can see in this picture, already after 30 seconds of illumination, the patch sticks nicely to the heart. You cannot even pull it out, and it is safe. We can also use gold nanorods as antennas with electroactive polymers to release or to have a better release of growth factors. In this case, it was SDF1, stromal cell-derived factor 1, which attracts stem cells and then allows them to build blood vessels. Finally, last series of projects that I'm going to show is what we call cyborg tissues, the integration of electronics with engineered tissues. And here what we've done, we created these nanowired mesh devices for electronics. We incorporated it into in this case, electron fibers to have scaffolding material, scaffolds with many, many nanosensors within. We cultured cardiac cells, and as you can see, we could see, this is just one electrode, but we could see the function of the tissue. We could add drugs like noradrenaline and see how the tissue reacts. So it was a nice publication and a good patent, but it wasn't enough because in this case we could only sense the function of the tissue, but if it was not working properly, we couldn't do anything. So in the next step, what we want to do is to allow the tissue or the electronics not just to record the function, but also to provide electrical stimulation or to allow us to release drugs. So for example, if the patient is not feeling well, the physician can log onto the computer and decide how he wants to remotely activate the tissue. For example, if he senses that there's not enough beats per minute, type on the computer, find electrical stimulation. If he senses that there's not enough oxygen, type on the computer, release pro-angiogenic factors to build new blood vessels. Or if there's inflammation, release anti-inflammatory drugs. And this is not just things that I imagine, these are actual things that we've done in this paper. And actually, we don't need the physician because we can write a simple code and this cardiac patch will basically regulate or control its own function. We created more flexible, more stretchable electronics in the clean rooms of university. We now can release up to 6 drugs in parallel inside the tissue. We can decide where we want to release the drug and when. And when the drugs are ending, I was often asked what's happening after 1 month, the drugs are not there, what can you do? And with a colleague in my department, we developed a way to modify the drug so that we can inject it into the circulation. It will find its way into the biomaterial and released over and over again. We did it in three cycles, and it works perfectly. So we can reload drugs into these materials. In collaboration with John Rogers from Northwestern, he creates his pop-up electronic devices. So we ship these electronic devices to Israel, and we incorporated the patient-specific materials with the cells, and we created these 3-dimensional, really thick tissues with electronics in all planes. We did some piezoelectric materials. Initially, we wanted to create materials that or scaffolds that will be used as capacitors. Put them on the heart and they will activate themselves, but it didn't work as we wished. But what we could do with that, we could culture stem cells, and once these stem cells differentiate to cardiac cells and started to contract, we could see it immediately on the computer how the signal is converted to electrical signal, as you can see. But the electronics that we used so far was made in the clean room with lithography. And the mechanical properties of these materials, of these electronics, were not as good, did not match the mechanical properties of the tissue. And if they're not matching the mechanical properties of the tissue, there will be an immune response, and eventually there will be rejection. And what we decided to do is to formulate the electronics to be printable and to have the same mechanical properties as the tissue. And this is what we did. So we printed the entire tissue with the electronics and the cardiac cells. Now we're doing it with the endothelial cells and the blood vessels together. And you can appreciate from this movie how soft this tissue is and how flexible it is. And basically, we can do the same thing. We can record and stimulate. We cannot release drugs with that. It's a different mechanism, and we're still working on that, but it will happen. We can create degradable electronics as well. Sometimes we don't need the electronics to stay for a long period of time. So we can do 2 things: either design the electronics to work, for example, for 3 weeks, or whatever we want, and then start to degrade, or to design the electronics to stay there and work properly until we give an external trigger, for example, with near IR, and then it will degrade. This is the pre-designed degradation. Show that it works, and after a while in the animals, after 3 weeks, starts degrading, and it's not functional anymore. Last slide that I'm going to show is now we're engineering the whole heart with electronics everywhere, so we can have electronic on the septum to transfer the electrical signal. We can have electrodes that release drugs on the left ventricle or whatever we want. So you can see here the recording and the stimulation. And with that, I will end up and acknowledge my group members, collaborators and funding them. I'm happy to hear any questions that you may have.